You are looking at a jar of honey. To you, it is breakfast. To a bacterium, it is a 5-part execution chamber. To infectious disease doctors, it is the one weapon that antibiotic-resistant superbugs have never beaten, not in all of recorded history. A food that is 80% pure sugar destroys the same bacteria that defeat our strongest drugs. How? And why has no bacterium ever adapted? Sugar is food for bacteria. Every biology textbook says so. Bacteria consume sugar, multiply, and cause infection. A jar of honey is a supersaturated sugar solution, 4 parts sugar to 1 part water. By every rule of microbiology, this jar should be a breeding ground, a petri dish filled with liquid candy. Instead, it is one of the most potent natural antibacterial substances ever identified. Ancient Egyptians packed wounds with honey. Greek physicians prescribed it for infections. Persian medical texts documented its power more than 1,000 years ago. Modern hospitals now stock medical-grade honey for burns and diabetic ulcers. Wound care centers use it on infections that refuse to heal. Here is the number that should stop you cold. Antibiotic-resistant bacteria kill 35,000 Americans every year. Methicillin-resistant Staphylococcus aureus. Vancomycin-resistant Enterococcus. Ciprofloxacin-resistant Pseudomonas aeruginosa. These are the names that keep infectious disease specialists awake at night. Honey kills all of them in laboratory studies, reliably and repeatedly. A food that is mostly sugar destroys pathogens that shrug off modern medicine. This is not folklore dressed up as medicine. It is peer-reviewed research published in the FASEB Journal and the Journal of Clinical Microbiology. The question stands: how does a jar of sugar kill what modern chemistry cannot? The answer is not one miracle compound. Honey contains more than 800 compounds, and its power comes from how they work together as a system. 5 distinct weapons, each one covering the weakness of the one before it. The first weapon is not chemistry. It is physics. Every bacterium needs water before it can do anything else. Not just any water. Loose water, free water it can pull across its membrane. Honey barely has any. The measurement is called water activity, and honey sits at 0.56. Most bacteria cannot grow below 0.91. That gap is a death sentence. When a bacterium touches honey, the physics takes over. The sugar concentration outside the cell is so extreme that water gets ripped out through the membrane. The process is called osmosis. The bacterium cannot stop it. It cannot mutate against it. Water flows out until the cell collapses like a punctured balloon. Picture a dry sponge on a wet counter. Honey is the sponge. It soaks up every drop of moisture from anything it touches. A jar of honey is a dehydration chamber for microbes. This weapon is universal. A bacterium that has survived every antibiotic ever invented dies the same way as one that has never seen a drug. Resistance genes mean nothing to physics. But here is the problem with this weapon. Pure honey kills by dehydration, yet diluted honey kills bacteria even better than pure honey. Water should weaken the first weapon. Instead, it makes honey deadlier. That should not be possible, and that paradox is the key to everything. Consider what honey actually faces in the real world. A wound is wet, blood is wet, saliva is wet. The moment honey meets living tissue, it begins to thin out. The dehydration weapon starts losing power immediately. And something else takes over. That something was hidden in plain sight all along. Honey is aggressively acidic. Its pH sits between 3.2 and 4.5. Most bacteria need a neutral range around 6.5 to 7.5 just to grow. That gap is a second wall, a chemical barrier they must break through before they can even multiply. The acidity comes from gluconic acid. Bees add an enzyme to the nectar, and that enzyme manufactures the acid over time. Here is the strange part. The enzyme lies dormant inside the jar, patient, waiting for a signal. The signal is water. The same water that disarms the first weapon arms the second. When honey gets wet, the sleeping enzyme wakes up and begins converting sugar into something far more dangerous than acid. Something that burns bacteria on contact. The bacteria that survive the dehydration chamber walk straight into a furnace. Water does not weaken honey. Water transforms it into a chemical weapon. The enzyme is called glucose oxidase. Bees add it to nectar while the honey is still being made. Inside the hive, it lies dormant, locked away by the low pH. Dilution changes everything. Water activates the enzyme. and glucose oxidase begins converting sugar into gluconic acid and hydrogen peroxide. The numbers are precise. Maximum production occurs at a dilution of 30 to 50%. That concentration generates 5 to 100 micrograms of hydrogen peroxide per gram of honey. Hydrogen peroxide shreds bacterial membranes and destroys DNA. But the weapon is not released all at once. It is produced slowly, continuously, over hours. Honey is a timed-release chemical weapon. The concentration stays high enough to kill bacteria, yet low enough to leave human tissue unharmed. It holds a steady, patient flame against infection. In 2010, a team at the Academic Medical Center in Amsterdam proved how effective this system really is. They tested medical-grade honey against the most dangerous bacteria on Earth: methicillin-resistant Staphylococcus aureus, ciprofloxacin-resistant Pseudomonas aeruginosa, vancomycin-resistant Enterococcus faecium. Every strain was killed by honey diluted to just 10 to 20%. A sugar solution matched to honey's exact carbohydrate makeup needed 40% to produce the same effect. Sugar alone could not explain the killing. Something inside the honey was doing the work, something that woke up only when the honey got wet. A study from the University of Waikato showed this system maintained its antibacterial activity for at least 24 hours. The slow release is the elegance. The concentration stays lethal for bacteria and gentle for tissue. But here is the problem with this weapon. It depends on an enzyme that is fragile. Heat destroys glucose oxidase. Pasteurization and commercial processing can strip honey of its peroxide-generating power. Heat a honey above 70 degrees Celsius and nearly all of this weapon disappears. And there is a deeper problem. Some bacteria produce catalase, an enzyme that breaks down hydrogen peroxide before it can do damage. In a deep wound, blood and tissue fluids can dilute the peroxide below effective levels. Yet certain honeys keep killing bacteria anyway, even when every trace of hydrogen peroxide is neutralized, even when the enzyme itself is destroyed. Manuka honey from New Zealand does not need this weapon at all. Manuka honey breaks every rule established so far. It comes from the nectar of New Zealand's Manuka bush. When researchers neutralized the hydrogen peroxide in Manuka honey, it kept killing bacteria anyway. When they destroyed the glucose oxidase enzyme entirely, the killing did not stop. Something else was inside this honey, something more stable than peroxide, something that did not need water to wake it up. In 2008, Thomas Henley's team in Dresden identified the compound and named it methylglyoxal. The concentrations they found shocked them. Manuka honey contains between 38 and 761 milligrams of methylglyoxal per kilogram. That is up to 100 times higher than any conventional honey. 100 times. That number explains why Manuka honey costs 100 times more than ordinary honey. The price premium is not marketing. It is chemistry. Methylglyoxal forms as honey matures. The Manuka flower's nectar carries a compound called dihydroxyacetone. Over time, that compound converts into methylglyoxal. That means Manuka honey grows deadlier as it ages. It does not decay. It arms itself. Once activated, methylglyoxal attacks bacteria on multiple fronts. It locks bacterial proteins in place, disabling their function. It interrupts cell division so bacteria cannot reproduce. It breaks down the protective wall around each cell. That it strips bacteria of their flagella, the whip-like tails they use to swim and invade tissue. A bacterium that cannot move cannot spread. A bacterium that cannot divide cannot multiply. A bacterium whose proteins are locked cannot function. But here is the paradox that breaks the Manuka story wide open. In 2026, Aston University researchers tested Manuka honey against solutions containing only methylglyoxal at identical concentrations. The whole honey consistently outperformed the isolated compound. The gap was especially wide against gram-negative bacteria, which shrugged off the pure methylglyoxal. Bacteria that carry a specific enzyme can partially neutralize methylglyoxal. Those that lack it are dramatically more vulnerable. That genetic difference proves methylglyoxal is central to the killing, not incidental. Methylglyoxal is not the whole answer. Something else in that honey is doing work that methylglyoxal alone cannot explain. That hidden weapon comes from the bee itself. It is an immune protein, and it has been sitting in every jar of honey ever opened. In 2010, the Amsterdam team identified the missing weapon. It is called defensin 1, an antimicrobial peptide produced in the hypopharyngeal glands of worker bees. The same glands produce royal jelly, the food that turns a larva into a queen. This peptide is the bee's own immune system, added to honey while the nectar is still being processed. It is the colony's immune system, weaponized and stored in every drop of honey. Quackman's team proved its importance with a brutal experiment. They neutralized the hydrogen peroxide and the methylglyoxal in honey. The honey still killed bacteria. Only when they neutralized defensin 1 as well did the honey finally lose its killing power. Every weapon had to be removed. The bee's immune system was the last line of defense. Defensin 1 kills bacteria by punching holes in their cell membranes. Picture a balloon being pierced with a needle. The bacterium's contents leak out and the cell collapses. This weapon is especially effective against gram-positive bacteria, including methicillin-resistant Staphylococcus aureus and Bacillus subtilis, the very strains that plague hospital wards. The jar on the counter contains the immune system of an insect, and it has been sitting in the pantry the whole time. A weapon forged over millions of years of bee evolution. Long before humans existed, bees were using it to keep their colonies alive. This weapon is not exclusive to Manuka. Every honey contains defensin 1. The cheap jar and the expensive jar share this defense. 4 weapons now stand revealed. Sugar dehydrates, peroxide burns, methylglyoxal dismantles, and defensin 1 punches holes. Each one covers the weakness of the one before it. Honey's antibacterial power is not an accident. It is a designed defense system. But bacteria have one more trick. They hide. In a wound, bacteria do not float freely. They build fortified communities called biofilms, slime cities with walls that antibiotics cannot penetrate. These hidden fortresses are why chronic infections resist treatment for years. Antibiotics bounce off the walls. The bacteria inside survive every assault. Watch what happens when honey reaches that city. It does not knock on the gate. It walks straight in. The first attack is a communication blackout. Bacteria talk to each other through chemical signals, voting on when to attack, when to multiply, when to build walls. Honey scrambles that signaling. The city loses its coordination. The orders stop coming. The network goes silent, wall to wall. The second attack is structural. Honey breaks down the glue that holds the biofilm together. The walls soften, they sag, and they dissolve. The bacteria inside become exposed and defenseless. The other 4 weapons flood through the broken gates. Dehydration, acid, peroxide, methylglyoxal, and defensin 1 arrive together. Research published in PLOS 1 showed honey disrupting established biofilms of Pseudomonas aeruginosa, Staphylococcus aureus, and Streptococcus pyogenes. The matrix binding them together simply came apart. A 2025 systematic review pooled 6 studies on medical-grade honey. The result was unambiguous. Honey significantly reduced biofilm formation and effectively disrupted established biofilms. The effect held against both laboratory strains and clinical isolates. Manuka honey killed biofilm-embedded bacteria more effectively than any single antibiotic tested. Rifampin, vancomycin, linezolid. All of them lost to honey. A bacterium that shrugs off every antibiotic on the shelf dies in the golden flood. Which raises the question that should unsettle the pharmaceutical industry. Why has no bacterium ever adapted to this weapon? Antibiotic resistance is the defining medical crisis of our time. 35,000 Americans die every year from bacteria that outsmart our strongest drugs. Methicillin-resistant Staphylococcus aureus alone kills more people in the United States than emphysema and homicide combined. Bacteria develop resistance because antibiotics attack a single target. One enzyme, one pathway. A single mutation can neutralize the drug, and the mutated bacterium survives to multiply. Honey does not work that way. The 28-day experiment is the proof. Researchers at the University of Wales Institute Cardiff exposed bacteria to sublethal concentrations of Manuka honey for 28 consecutive days, generation after generation. The bacteria never developed resistance, not once. A separate study confirmed the result. Bacteria already resistant to multiple antibiotics showed the same vulnerability to honey as bacteria that had never seen a drug. Antibiotic resistance provided zero advantage. Nurul Coxetin, a research fellow at the University of Technology, Sydney, put it plainly. Bacteria quickly learn how to fight off antibiotic drugs. They do not have honey resistance. Every antibiotic ever invented has been defeated eventually. Penicillin lasted decades before resistance spread. Honey has been used for thousands of years. No bacterium has ever adapted. Not in a laboratory, not in a wound, not in all of recorded history. Why? Because every antibiotic ever invented hits one target. A single mutation rewrites the lock and the drug no longer fits. Honey attacks with 5 weapons at once: dehydration, acid, peroxide, methylglyoxal, and defensin strike together. To survive honey, a bacterium would need to mutate against all 5 simultaneously. The probability is effectively 0. And then comes the part that should rewrite how medicine thinks. Honey does not just kill resistant bacteria, it makes them vulnerable again. Manuka honey increased the sensitivity of Staphylococcus aureus to linezolid, an antibiotic the bacteria had learned to resist. The honey reopened the door the bacteria had locked. Studies show honey restores the power of drugs the bacteria had already defeated. Medical-grade honey now sits in wound care centers as a first-line treatment for burns and diabetic ulcers. Infections that shrug off vancomycin and rifampin are being cleared by a food. The pharmaceutical industry spent decades and billions engineering single-target weapons, and every single one fell anyway. Bees built a 5-weapon system over millions of years. Evolution has not touched it. There is one final difference between honey and every drug ever made. Antibiotics kill without discrimination. They destroy the infection and the protective microbiome with it. Honey does the opposite. It kills pathogens while feeding the bacteria the body needs to survive. It is a prebiotic. The complex sugars reach the colon intact and fertilize beneficial strains. Bifidobacteria and lactobacilli thrive on what honey provides. A 2024 University of Illinois study proved this in humans. 66 adults ate yogurt with clover honey for 2 weeks. The honey protected the probiotic bacteria through the acid of the stomach. More good bacteria survived the journey. Honey does not wage war on bacteria. It prunes the garden. Destroy the dangerous while nourishing the essential. No pharmaceutical antibiotic has ever achieved this. The jar in the kitchen holds a weapon system medicine cannot replicate. 5 weapons in one jar: dehydration, acid, hydrogen peroxide, methylglyoxal, and defensin 1. Each covers the weakness of the one before. Dehydration fails when honey gets wet, and peroxide wakes to take over. Peroxide dies in heat, and methylglyoxal steps in. Methylglyoxal alone cannot explain the whole story, and defensin 1 fills the gap. The final weapon is not a compound. It is selectivity, a targeting system millions of years in the making. Honey kills the bacteria that defeat our strongest drugs. It feeds the bacteria that keep us alive. After thousands of years, no bacterium has adapted. Not in a laboratory, not in a wound, not in all of recorded history. That is not a food. That is a message from evolution itself.